Hello everyone. Today we're going to learn some aspects related to hormone receptor interactions. Our learning objectives include classify the various types of hormones, describe the hormone secretion, transport and clearance from blood, classify the various types of receptors, describe hormone receptor interactions in terms of upregulation and downregulation. Upregulation and downregulation, define secondary messenger and list the various secondary messenger systems, differentiate between genomic and non-genomic effects of hormones. And this is going to be a elaborate lecture which I will be breaking into pieces. Some portions will be covered in this video, later in the next video. So let's start. Before moving on to the complexities of this topic, first let's learn some important jargons or glossary of terms. Catecholamines. They are a group of amines that include epinephrine, norepinephrine, and dopamine and have important physiological effects as neurotransmitters and hormones. Then downregulation. The decrease in the number of receptors in response to a high concentration of circulating hormone is referred to as downregulation. Then ligand. It is derived from the Latin word ligandus, meaning to bind. A ligand could be an ion, a molecule, or a hormone that binds to another chemical entity or a receptor to form a target complex, to form a larger complex. And upregulation, an increase in the hormone receptor number as a consequence of a sustained low exposure to a particular hormone is called upregulation. Okay, now first topic, chemical structure and synthesis of hormones. Hormones can be classified based on their origin or by their chemical structure. Three general classes of hormones exist, which are the proteins and peptides, including hormones secreted by the anterior and posterior pituitary gland and pancreas, insulin and glucagon, the parathyroid gland, parathyroid hormone, and many others. P for hormones secreted from posterior pituitary gland, pancreas, and the parathyroid gland. Okay, PPP. And just remember as protein and polypeptide. Parathyroid, pancreas, and pituitary. Pancreas, parathyroid, pituitary. Okay. Then steroid secreted by the adrenal cortex. Next set proteins and peptides. Then next steroids secreted by the adrenal cortex, cortisol and aldosterone, and ovaries, estrogen and progesterone. The testes, testosterone, and placenta, estrogen and progesterone. The next derivatives of amino acid tyrosine. Derivatives of amino acid tyrosine secreted by the thyroid, thyroxine and triiodothyronine, and the adrenal medullae, epinephrine and norepinephrine. So three categories: proteins and polypeptides, steroids, then derivatives of amino acid tyrosine. Just don't forget which is that amino acid, that is the tyrosine. Okay, derivatives of amino acid tyrosine secreted by the thyroid, thyroxine and triiodothyronine and adrenal medulla, epinephrine and norepinephrine. Okay, so we learned three categories, general classes of hormones. Peptide and protein hormones are stored in secretory vesicles until needed. Most of the hormones in the body are polypeptides and proteins. These hormones range in size from the small polypeptides with as few as 3 amino acids, thyrotropin releasing hormone, to proteins with almost 200 amino acids, growth hormones and prolactin. In general, polypeptides with 100 or more amino acids are called proteins, and those with fewer than 100 amino acids are referred to as peptides. Protein and peptide hormones are synthesized in the same fashion as most other proteins. Steps in the synthesis of protein or polypeptide hormones are as follows. First, synthesized as large biologically inactive protein, pre-pro hormones. Pre-pro hormones. Okay, pre-pro hormones cleaved to form smaller prohormones. prohormones. Then that pre-hormones are then cleaved into smaller prehormones, which are situated in the endoplasmic reticulum. They are present on the ER. Okay, they are on the ER. Then transferred to the Golgi apparatus for packaging into secretory vesicles. Enzymes in the vesicles cleave the prohormones to produce smaller biologically active hormones and inactive fragments. So first synthesized these protein or polypeptide hormones are synthesized as large biologically inactive proteins, which are called pre-prohormones, cleaved to form smaller prohormones in the endoplasmic reticulum, then transferred to the Golgi apparatus for packaging into secretory vesicles. Then transferred to the Golgi apparatus for packaging into secretory vesicles. So, pre-prohormones then cleaved to form smaller prohormones in the endoplasmic reticulum, then transferred to the Golgi apparatus for packaging into secretory vesicles. Enzymes in the vesicles cleave the prohormones to produce smaller biologically active hormones and inactivate fragments. Then the vesicles are stored within the cytoplasm until their secretion is needed. Secretion of the hormones as well as the inactive fragments into the interstitial fluid or directly into the blood serum occurs by exocytosis. Okay. When in many cases the stimulus for exocytosis is increased by cytosolic calcium concentration caused by depolarization of the plasma membrane. In other instances, stimulation of an endocrine cell surface receptor caused increased cyclic adenosine monophosphate and subsequently activation of protein kinase that initiates secretion of the hormone. The peptide hormones are water soluble, allowing them to enter the circulatory system easily where they are carried to their target tissues.